In this part, we are going to continue about the process, applications and limitations of bioaugmentation. So as we have already discussed, bioaugmentation is the process where we add organisms at the site of the pollution to increase or to start the process of bioremediation of the pollutants like heavy metals, azo-dyes and hydrocarbons. So this process works on certain mechanisms. Two mechanisms we have already discussed. Now we will discuss the third mechanism, that is bioaccumulation. Bioaccumulation involves microorganisms taking up and accumulating the pollutants within their cells. This mechanism reduces the concentration of pollutants in the surrounding environment by sequestering them. So in this mechanism, organisms take up the pollutants from the surrounding environment and they store those pollutants inside the cell, hence making the pollutants immobilized from the environment. How it works? First is pollutant uptake. Microorganisms can absorb pollutants from their environment and store them inside their cells. This often occurs through passive diffusion or active transport mechanism. The pollutants can be stored in the form of certain salts or they can be stored in the organelles like vacuoles or lysosomes where they cannot harm the cell itself. For example, heavy metal accumulation occurs in certain bacteria like Bacillus or Pseudomonas species. They can accumulate heavy metals like cadmium, lead and mercury. These metals are sequestered within the microbial cells, reducing their availability and toxicity in the environment. This is applied for heavy metal removal, which is used in the treatment of contaminated water and soil to remove heavy metals. And it is also used in the bioremediation of mining sites where microorganisms are employed to clean up the metal mining sites. The last mechanism which is used by the organisms for the process of bioaccumulation includes synergistic effects. Synergistic effects occur when different microorganisms work together in a consortium to enhance the overall degradation process. So here different organisms come together and they enhance the effect or efficiency of the process. So the collaboration of different organisms can involve complementary metabolic pathways or mutualistic relationships where both the partners are benefited. It works by complementary rules where different microorganisms can perform sequential or complementary steps in the degradation process. For example, one microbe might degrade a pollutant to an intermediate which is the starting material for the second organism. So second organism will take up that material and convert it to the molecule which is less harmful or non-harmful. For example, lignin degradation. In the degradation of lignin, which is a complex plant polymer, white rot fungi and bacteria work together. The fungi breaks down lignin into simpler compounds which are further degraded by bacteria. Bacteria cannot directly degrade the lignin. Applications. This is used in case of complex pollutant degradation. It is used in bioreactors or bioremediation projects where complex contaminants like mixed industrial wastes are involved, which are in need of different metabolic pathways provided by different organisms. Also, this method enhances soil remediation. Consortia of microbes can be used to treat contaminated soils or more effectively by targeting different pollutants and degradation pathways. Benefits and challenges of bioaugmentation include increased efficiency, especially when we are discussing about synergistic interactions. Synergistic interactions can enhance the overall efficiency of the pollutant degradation. And also, in this process, as different organisms are used together, broader range of pollutants can be degraded at the same time by complementary actions of the organisms which are involved. The challenges in this process are complexity in design. Creating effective microbial consortia requires careful design and understanding of microbial interactions. The organisms which are used together should not compete with each other. They should act synergistically. And also, stability and management is also one of the challenges, as maintaining the stability and activity of microbial consortia in the field can be challenging, because as the concentration of the pollutant will change in the polluted site after take-up of those molecules by the organisms, the population will vary. This might lead to some complications in maintaining the stability. The application of this process involve environmental remediation, where the organisms are used for cleaning up contaminated soils, sediments and waters, such as those affected by oil spills, heavy metals or pesticides. Also, this can be used for remediation of wastewater. It enhances the degradation of organic matter and pollutants in sewage and industrial water. The method can also be employed in agriculture, that improves soil health by adding microbes that can fix nitrogen, decompose organic matter, solubilize phosphate, or suppress soil-borne pathogens. This method is also used in industrial processes to optimize the processes such as biotransformation in the production of chemicals or recovery of valuable resource from waste material. Benefits are efficiency. This method can significantly speed up the degradation of pollutants compared to natural processes. Also, the method is very specific, that allows the targeted treatment of specific contaminants, and it results in environmental restoration, as it helps in rehabilitating contaminated environments to make them suitable for reuse or habitations. The challenges involve survival and competition. As in synergistic effect, we introduce different organisms together. There are chances of the organisms of competition with each other, and some organisms may struggle to survive and compete with native species, potentially limiting their effectiveness. It also increases the cost of the process, as there is need of isolation, culturing, and application of specialized microorganisms. It might affect the environment, as there is a risk of unintended ecological impacts, such as the introduction of invasive species or alteration of existing microbial communities. Also, there are challenges including regulation and safety. The use of genetically modified organism, which is a common practice which is involved in bioaugmentation, this is subject to strict regulations, and there are concerns about the potential risk to human health and the environment. Applications of bioaugmentation process include environmental remediation, mainly soil decontamination and water treatment. Soil decontamination can be achieved by bioaugmentation, as bioaugmentation uh, is the application of different organisms to the polluted area. So, if the soils are contaminated with hydrocarbons, heavy metals, or chemicals like pesticides, insecticides, chemical fertilizers, then those molecules can be degraded by using certain bacteria or fungi. After addition of these organisms, the pollutant molecules will get broken down into simpler non-toxic molecules. Also, it can be used for water treatment. In wastewater treatment, plants microorganisms can be introduced to enhance the breakdown of organic matter and pollutants. This includes both industrial wastewater as well as municipal sewage. Bioaugmentation also helps in agriculture, as it enhances the soil health. Certain microorganisms can improve soil fertility and plant health by decomposing organic matter or fixing nitrogen or suppressing plant pathogens. Also, there are some organisms which can solubilize phosphate. Biopesticides. Microorganisms can be used to control plant disease or pest, reducing the need for chemical pesticides. In industrial processes, it is used mainly for biotransformation. Bioaugmentation can be applied to industrial processes to enhance the efficiency of biotransformations, such as converting waste products into valuable byproducts or energy sources. The waste material can also be used to grow organisms in the form of single cell proteins. Mine site rehabilitation, metal recovery, microorganisms can assist in the recovery of valuable heavy metals from ore or mine tailings. They can also help in the detoxification of metal rich waste, and these metals can be desorbed and recovered and reused. Limitations. The main limitation is environment compatibility. Survival and activity. The introduced microorganisms might not survive or be active in new environment due to unfavorable conditions such as temperature, pH or oxygen level. Suppose for example we want to bioremediate the oil spills from ocean and we are adding some organisms, so we need the organisms which can grow in high salt concentration. So the choice of organisms need to be done very smartly. Also, the bioaugmentation process has certain ecological impact as we are adding the organisms which are not native of that place. So they may harm the surrounding organisms, or environmental conditions may also change due to addition of these foreign organisms. So there is a risk of unintended ecological consequences such as the introduction of invasive species or disruption of existing microbial communities. Cost and complexity. Economic feasibility is less because for selecting the organism, for studying their characteristics, then culturing them and applying them, at every stage of this there is requirement of raw material which increases the overall cost of the process. The cost of isolating, culturing and applying specialized microorganisms can be high, making bioaugmentation economically challenging, especially in low value or large scale remediation projects. And technical challenges are also involved as developing effective formulations and delivery methods for microorganisms can be complex and it requires specialized knowledge. Regulatory and safety concerns. The use of genetically modified microorganisms, which is a common practice in this method, is heavily regulated in many regions, which can limit their application or delay the projects. Safety. There are concerns about the potential for unintended spread or impact of the genetically modified organisms, which could have unforeseen consequences for human health and the environment. Long-term efficacy. The organisms which we are applying at the polluted site should show efficiency consistently. Persistence is one of the factors. Microorganisms introduced through bioaugmentation might not persist long enough to complete the desired degradation or transformation process. The degradation product sometimes may be harmful or toxic than the original compound, raising concerns about end products of the bioremediation process. So there is a need to keep check on the byproducts which are getting formed as a result of bioaugmentation process. Interaction with native microbiota. There may be two types of interactions. One is competition, which is not very desired for the process of bioaugmentation, where the introduced microorganisms will compete with or to be outcompeted by native microorganisms, which can limit their effectiveness. Synergistic effect is one of the factors. If this is achieved, then the desired outcome is easily achieved. There needs to be a careful balance of the microbial interactions, which can be difficult to manage. Successful bioaugmentation treatments depend on the use of inocula consisting of microbial strains or microbial consortia that have been well adapted to the site which is to be decontaminated. Foreign microorganisms which are present in the inocula have been applied successfully, but their efficiency depends on the ability to compete with indigenous microorganisms, predators, and various abiotic factors like pH, temperature, or oxygen concentration. Factors affecting proliferation of microorganisms used for bioaugmentation include the chemical structure and concentration of the pollutants, the availability of the contaminant to the microorganisms. That means if the contaminant gets adhered to some particles like soil particles, then it becomes less available for the organisms for taking for its bioremediation. The size and nature of the microbial population is also one of the crucial factors, and also the physical environment should be taken into consideration when screening for microorganisms is to be done. Bioaugmentation involves the introduction of microorganisms isolated from the contaminated site, from a historical site, or carefully selected and genetically modified to support the remediation of petroleum hydrocarbon contaminated sites or heavy metals or dyes, whatever type of pollutant is present. Here we need to either assume that these organisms, as they are present in the natural site, they might have the capability to degrade the pollutant, or we need to confirm this using some laboratory experiments. So if we can use the organisms with proper knowledge, the bioremediation can be achieved by bioaugmentation.